And after you find the highly variable genes and you define the normalization, scaling, what you do is the principal component, right? So what is the principal components? As you know that this is the very typical dimension reduction algorithm, right? So they will find the main axis of variation and denoise the data. So as you know that the main axis represented by principal components, right? And this is very commonly used in single cell analysis because the PCA give you reduce the dimension, as I mentioned, the principal component, and this principal component is really useful way to describe the relationship between the cells, between the genes, and that will give you the cell types automatically, right? So PCA plays a pivotal role in single cell analysis by reducing the high dimensional gene expression data and summarizing to the lower dimensional space, and it preserves the most significant variation in the data set. So what it's doing, dimensional reduction, single cell data set are typically high dimensional because it has the multiple cells, multiple genes, right? So basically same meaning. And it's really hard to make a good visualization into the 2D space, right? So we have to reduce the dimensionality of the data set first and into the orthogonal axis. And we can easily describing the linear combination of original genes. And the next thing we can do is the variance maximization. So PCA identifies the principal component in such way that the first principal components capture the maximum variance, as you know that from the PCA. And the second principal component can capture the second, highest variance dimensions as well. But the problem is, if you think about the single cell status in your data set, there's no two-dimensional, so there's no two relationship within a single cell because, cell types is already more than two, most of the case. And also the cells are highly, dynamic across the different states, right? So you can expect multiple dimensions we can get from the single cell data set, right? So this is again why we need PCA because we want to, capture these variants at the most likely way, right? And the third thing is the noise reduction and feature selection. So PCA can remove noise and focus on biological relevant variation in the data set. And it is discarding the less informative principal component, and PCA effectively select the subset of genes contributing to the variation by the biological phenomena. And the feature selection aspect can simplify downstream analysis and making computationally more efficient and biologically more interpretable. So visualization is the, you can use the 2D or 3D scatter plot or using the Umap. We're going to go through in later sections. Okay, so how we can do the PCA? So we still use this CAMP library and TL module. So we're not going to use the PP. So PP is the pre-processing, and TL is the you use for the dimension reduction. And within this module, you use the PCA function. I mean, very, very easy, right? And you can put the A data on here, and you use the highly variable genes, and you use SVD solver. This is which algorithms, especially you use for the computation. So once you've done that, then you can see PCA loading, right? So this is PC1, PC2, and PC3, right? And each of the points you can see highly variable genes. So first thing is, can you see that? You probably see from the notebook I provide you. This is the LYZ, and next one is A100, A9 something, and you have this order. So as you can see here, you see the number of genes, they make a distinction between these two axes, right? So the values on here, it doesn't really have meaning in biological system. It's more like a eigenvalues, right? So one values are heading towards this direction with these genes, Another direction is composed by these genes. So you can see that this gene and these genes are probably working in different ways, right? But anyhow, PC1 has bit of variation between certain set of genes, so which means that these genes are likely distinguish the cell types within our data set. Okay, so one of the genes we can go for further is the CST3, probably appear somewhere in these plots. And you can see PC1 and PC2, you can see that the cells on this part, they have high expression of CST3 genes and lower expression on this end, right? So you can see that the genes can distinguish the cells within the data set. So that will ultimately forming the cell types, right? Or cell clusters, basically same meaning. Okay, so now we can inspect the contribution of single pieces to the total variance in the data set, right? So as we know that the PCA give you highest variance at the very first principal component, right? So as you expected, PC1 has the highest variance, PC2 second highest, PC3. But the question is, how we can get the sort of very high variance pieces up to certain numbers, right? So we can just say that PC1 and PC2 give you highest variance and rest of them are meaningless. Or you can say that PC1 to PC20 are highest variance and rest is marginal. So you have to make sure that, how many pieces are actually having, meaningful, which means that some, degree of variance within the data set. So as you can see here, PC1, lots of variance, PC2, it's a lot, PC3, 4, 5, 6, and up to somewhere this point, right? So you can see that PC1 and PC2 are very different. They have deviation between two. So we say PC1 is the top variance, PC2, PC3, a little bit similar, but compared to PC5, it's a lot, right? And PC7 and 8 a lot, but PC8 and 9 is not that great as much as you see PC7 and 8. Then you can see that the deviation between the nearby pieces, they getting lower and lower, right? So up to this point, they cannot explain that much, right? So you have to set up certain pieces you want to use for finding clusters, finding Umaps, right? It's really arbitrary. So we normally say no more pieces change of their variance. We choose those numbers. So I would go like a PC20 or PC maybe 30 something, but definitely not PC4 or PC3 because PC5 also have bit of meaningful variance as well. Okay, so after you run the PCs, if you look at the A data, it has another information. So it has OBS, it has a VAR, it has the UNS, but now we have OBSM VARM, which is the matrix version of observation, matrix version of VAR. What they containing is the PCA or the pieces information. So these things give you some information, right? Okay, so once you get the PCA, what we're going to do next is compute the neighborhood graph. So why we care about the neighborhood graph? Because you have the PCA from your single cell data set, but now you want to make sure, how many cells are they have the cluster relationship and how many cell types we can explain by these pieces. So this time, we have very specific computational algorithm called neighborhood graph to define the boundary of this cluster relationship. So how we can get that information? This is, we use the same function called SCPP neighbors, and we can say that we want to use the PC10. Okay, so the tutorial, they want to use the PC10, so up to here. I would go PC20, but yeah, that's fine. So PC10, and you want to identify neighbor 20, right? If the, I mean, these things can, also the arbitrary way, you can adjust the numbers. You want to find the best parameters explaining your biological experiment. So it's really up to you, but just go with very, expected numbers like a 10 or 20 is sort of very common numbers. Okay, so I'll skip, just explaining this bunch of text because, I mean, visually you can think about what we are doing now, right? So you basically find the neighbors using this PCA, then you basically embedding your neighborhood graph into the two-dimensional space. So that space you want to use the Umap, which is to visualize your PCA. So this is again, you use the function called Umap, literally Umap, and put the A data. Then you have 10 PCA, right? So what that means, you have the PC1, oh, sorry, I'm a bit lazy. So PC1 and PC2, and you have dots like this, right? And you have PC1 and PC3, you have PC1 and PC4. You can draw this graph up to like a 10 and 10, right? So you draw like 100 two-dimensional plot, right? So what UMap's doing is basically collapse all that information into the two-dimensional space, right? So you can basically put the pieces with your neighborhood graph and within an A data, then you can basically plot this graph like this. Then you add on some genes or anything you want to visualize within your A data. So this case, we put the CST3 genes expression on here, right? But the point is that UMap is basically collapse that information within this space, right? So what is the different things with the previous one? So if you look at here, so PC1 and PC2, right? But since we use the up to PC10, you have like around like tens of the graph like this. So we merge all the PC10 information into the UMap. And afterwards, you use this UMap and the neighborhood graph, then you start clustering, right? So this time, we can use many different clustering methods like a k-mean clustering or different ways of clustering. But single cell analysis, we have two famous clustering method. One is the Lubanian method, and the other is the Leiden method. You don't really have to remember, but just go with the Louvain. And Louvain method, we put the A data, and you set up the seed, the 42. Then you set up the resolution parameters, how you want to go detail of them. So we set up the resolution. I actually want to show you how the resolution is working. Otherwise, it will be very confusing. So let's load this one again. Okay, so highly expressed filtering mitochondria. Okay, this is working. And loop transformation, highly variable genes, and highly variable genes. Then the scaling. Then the PCA. I have to do finding neighbors and UMap Great. Then here we go. So we have the Rubenian TL, and I think we should go this one first. Oops, I knew that these things can happen. So color. So I need to explain what I'm doing. So once you ran the Rubenian, then you see the result is came in. So Rubenian here, right? So you see A data OBS, the clustering result stored on here, right? So what I want to do is I want to color this Umap graph by Rubenian cluster. So this is the numbers, right? So if you run it, you can see that the cluster 0 on here, cluster 5 on here, cluster 1 is here. See? So this is how the clusters are made. Then what I want to show you is, so we use the resolution 0.5. We want to see high resolution than 0.5. We can see that how these things is working. So let's try 1.5. We see the extreme things. Okay, can you see the differences between two? So this is the Rubenian resolution 0.5. You see the 7 clusters. If you use 1.5, you have 12 clusters. So if you see the cluster number 0, which is the highest number, is split into 3 different clusters, right? So why these things possible? We explain the biological name of these clusters later, but what I want to say here is the cells are not the same to each other. Cells in a biological system, cells in you, cells in a human, cells in animals, cells in plants, they are not same to each other, even though we say this is the T cell, this is a neuron, this is whatever adipose, but they are actually very different. They're different characteristics, and they continuously changing. Even you're listening to this YouTube video, even if you're listening to my explanation, even I say my cells are continuously changing, right? So unsupervised clustering, one of the characteristics is that they have continuous boundary to each other. This is not really like a two different binary distinctions, right? So you see that these large clusters can divide into 3, but even it can divide into further, right? See, you can see further. So which resolution numbers are correct? As I say, it's an unsupervised learning. It's up to you. So if you take out this cluster 0 and it explains your hypothesis correctly, then you go with this. But if these things is not enough for your hypothesis, you can go with this. So that's why I mentioned in the previous sections why we kept the A data row, because you might want to repeat these steps many times, right? Because unsupervised learning is the exploring way you want to ask yourself to have the answers for your hypothesis or your research questions, right? So that's why parameters is really arbitrary. It's up to you. Okay, so after that, what you want to do, you want to think about the biological meanings of these clusters, right? So you want to find the genes highly expressed in these cells or in these cells or in these cells, right? So how we can do that? We can use the Rubenian, and we use the t-test to see the expression differences. And we use the correction method by Bonferroni because we're testing multiple hypotheses, right? So if you rank genes group, then you see the top 25 genes on a high expression. So first gene is this one. So this is highly expressed in cluster 0, and IL-32 highly expressed in cluster 1, Is this really rare? I actually want to go here again. Then IL-32 highly expressed in 1. Is this really true? So we can say IL-32. Oh, sorry. Probably this one. Okay, so if you look at this, this is the cluster 1. Damn it. So here is cluster 1, right? Cluster 1. And IL-32 is highest expression here, right? And it's highest expression on cluster 6 too. Is that really true? Yeah. IL-32 highest expression in cluster 6 as well. Okay, so this is how we define the highly expressed, differentially expressed in certain cells, right? Or you can compare different way. We can use the Wilcoxon test. It's really up to you. So some of the data set we have to use t-test, some of tests we have to use the Wilcoxon. It's a little bit tricky because the data are really different to each other, and some of the tests are really sensitive, or some of the tests are really specific. So scientists still debating which statistical method good for these finding marker genes, but people generally use the Wilcoxon test. So you can just go for your things too. But I just want to empathize the results can be a little bit different by different statistical procedures, and it's still, under developing. Okay, so another way we can try is log regression. That is also another method we can try instead of Wilcoxon test or t-test. And afterwards, we explore some of the genes, and we use these the genes with the legend known for certain cell types. So let's say IL-7R is non-gene marker for CD4 T cell, CD14 is monocyte, and MS4A1 B cells. All of that information, if you use this information, then you can judge which markers are represented in each cell types. You can go with the manual way. Then you see cell type 0 is really do something, or cell type 1 is doing something. So it is also sort of the manual way you can think about the relationship. Okay, so this is all that related information, so you can read about that. Then lastly, once you get all that information, we can use the pathway analysis. So what is the pathway analysis? So we have the bunch of genes are specifically expressed in a certain cell type, right? And these cell types are very common in in certain cell clusters because they're explaining certain biology underlying the cell types, right? So let's assume that we have here, sorry. So Ruben group 0 is explained by IL-7R, which we assume that this cluster 0 is the CD4 T cells. So we are expecting there must be CD4 T cell pathways are highly expressed in cluster 0, right? So likewise, we can think about these pathways within each clusters, right? So how we can do pathway analysis in your single cell data set. So one thing you have to remember is you need some pathway data set to think about this pathway enrichment, right? So most of the case, what we used is the molecular signature database. So this is the database I think I put the link on here. So if you click this one, okay, I want to go there. So this is the M6DB collection. So you can see different gene sets on here, right? So some of gene sets are explaining the hallmark of some biology, so very representative biology. So if you browse the 50 gene sets, it has the adipogenesis, allograft rejection, immunology, and androgen response, angiogenesis, and up to whatever the Wnt beta-catenin signaling or something like that. So all of that information you can get from this data set. So what I want to say, if you want to compare the pathway in your single cell analysis, you need some data set to represent those information. And then afterwards, you have to set up the null hypothesis in your genes enrichment. So basically, meaning that if you try some genes within a certain pathway, your expectation is those genes are not changed, and it compared to background gene expression. So what that means is that if you look at the cluster 0, which is the T cell type, then your null hypothesis is if you want to test the T cell pathway genes, these genes have the same gene expression as you see in other gene sets, in other genes in your cluster 0, right? Then you measure statistics. Then if that statistics procedure reject your null hypothesis, then you can say that the T cell gene expression is highly perturbed, highly altered in your data set, in your cluster 0. Then you can think about the related pathways in cluster 0, right? So there are many different ways we can test the gene pathway analysis. So the most famous thing is you can try the GSEA, gene enrichment analysis, or you can try the t-test, or you can use the differential gene expression genes from the differential expression test. So these are all the things you can try, and I summarize different methods in here so you can have for your information. Okay, so what we can do first, we are going to pathway enrichment test using the decoupler. This is very easy package you can try by yourself. So first thing is you have to pip install decoupler. Then you install some of the related information. Then you import the decoupler. Then first thing is you have to load the GMT, the genset pathway file for the C2 one in an MSIG DB. So this is the file we get from here. So C2 file. So you can click this one and you can download the file, right? But you can also try Hallmark. Also, you can try other pathways. So if you do the assignments, please use the different pathway GMT file. Then that will give you more variable results than just C2 pathway. Okay, so we set up some functions to make more easier way to do it in your analysis. And after that, you load the data into the decoupler format. Then afterwards, you can check which data set you add. So C2 file is basically the reactome file. So I put the reactome here. Then you can see the gene symbols and which pathway collection you are getting. Then you see which gene sets they are involving, right? So some of the gene MFF is the megakaryocytes development something, and all of the information you can see from here. Okay, so how we run the GSEA. So we set up which genes we're going to use, and we use this is just an example. We use the Rubenian cluster number 0. Then we subset the gene just for the cluster 0. Then we run the which test we are using? GSA. So we're getting the information. Then after we run the run GSA on here, then this is sort of the results. So you see the scores and p-value. So which pathways are really highly, related, enriched in this cluster 0. This is the neutrophil degranulation. So, and the second is MHC class 2, antigen presentation. and last one is the carbohydrate something. So we can use the seaborne, then you can plot this information. These things has to be appeared on here, but somehow my Colab is not really working. So please make sure your things is working in your assignment. So, I mean, it has to be, I think it's some marginal issues. It has to be coming on here. Okay, so we looked at all the information about the single cell analysis from the RNA-seq to QC and clustering and find the marker genes and pathway analysis. So this is everything about the single cell analysis, and you can do sort of the basic things by yourself from here, and this will help you understanding and reading the single cell RNA-seq papers. So, I mean, please try this by yourself. This is really easily done by Colab, and this is freely you can do with your Google account. So you don't need to pay anything for that, because Google Colab give you the free credit for running some computational programs. So this tutorial is basically runnable within this free credit. The next thing I want to highlight that is the assignment. So we have the assignments for this tutorial. So what you have to do is you basically write down the analysis by your data, right? So what I want to ask you is, I don't want to ask you the private data of your experiment or your research project. This is for finding data set from public database, right? So you first find the data set for your analysis, then you write down the single cell analysis as you see in this tutorial, right? You can change a little bit order by yourself, but that's fine. But you have to write down these things by your own data set you search from Geo database. So if you go to this database, Geo, then you can search something from here. So I want to search mouse brain. You can search that up, the mouse brain and 10x. Then that will give you some of the information. So here is the persistence of homeostatic sleep derived is encoded by blah, blah, blah. And it looks like a 10x single cell data set. I don't think it's really a good thing. Maybe this one. Okay, so this is good. So this is the transcriptional reprogramming, blah, 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 anesthesia, wild type, Alzheimer mouse model. And if you look at the description, and you can see that there is a file, right? So if you click the HTTP, then you can download the matrix file. So you can load the data into your system. Then you can try by yourself. So please find the data set by yourself, and it has to be unique. My expectation is there's no one can have the same geodata to your friends or to your peers or to your classmates, right? So I give you the Excel sheet on Google Spread. Then you have to upload your things. The first one coming first, right? So then you can basically find your data set first. Okay, so this is basically the description. I need to change these things further. It has to import it in the scan p package, right? the other method. Okay, so that was it. One thing students might be a little bit confusing is you need to know about the code. What I want to say is, where am I? Data sets. Input. I'll give you an example. I don't think this is the one. I think this is the one. I think this is the one. Import. Okay, so the things like, as you see here, this is the MTX file. This is a little bit different from H5AD we used in a tutorial. So that case, oops, you have to go with the different ways of loading the data set. I'll give you like this. MTX file, something like this. Your file. So import. You can, you can figure out by programming, but sorry, this is not really working anyway. So this is for your reference. Okay, I don't want to keep this one as it is. So, okay, and then the evaluation criteria is a total 100%. So data overview is the 20%. So you describe what you want to do, and you describe the GEO information and background and research question. Then second thing is the data processing and exploratory data analysis. And third one is the differential expression analysis. And last one is the biological interpretation. So this is total 100%. Okay. And submission has to, complete it in a Colab notebook, and you don't need to provide the additional output file, just the Colab notebook, download into IPy book format. Download your notebook file with output. The file should be student's number, name, that IPython notebook. Okay. So example is 1, 2, 3, 4, 5, 6. Maria, Kim, IPyMB. Okay, so that is the expectation. And there is no score for the late submission, so please keep in mind that. So how we can download this IPython notebook? You can click the file, and you can click the IPython download, right? So let's download this file. Then if you run it, if you open it, if you open it, you can actually see, like this, right? You can actually see, you can actually see the outputs like this, right? So plots, all the plots like this, okay? So your notebook should contain this output into your research, right? So please keep in mind that. I need to tell you tips. If you want to check your iPy notebook file, you can open it by MS Visual Studio. Visual Studio Code. Okay, so this is the program I opened. Okay, so that's everything, and hope you enjoy the tutorial, and hope you submit your assignments on time. And if you have any question, then feel free to ask me anytime. Okay, so that's everything. So see you next time.